Now that we've learned a little bit about gene function as an individual protein or gene, we'd like to look at gene function in terms of larger context and other proteins. And for that, we're going to look at pathway information. Here I'm back on the Entree gene P53 page. We're going to look at pathways, and here are a lot of pathways that P53 partakes in. I think we'll focus in on P53's role in the cell cycle. Here's an intermediary page, but with some description about the way the pathway is managed. But now we'll click on the link, and this takes us to one of the two primary pathway repositories, and KEGG, K-E-G-G, stands for the Kyoto Encyclopedia of Genes and Genomes, maintained in Japan. And now we look at the cell cycle pathway. We see P53 as being sort of a node here. We see some downstream proteins that it's interacting with, and we see that this would inhibit cyclin D. Remember, the cyclin proteins are key regulators of the cell cycle, and in fact, cyclin D is involved in the G1 to S cell cycle stage progression. Here's the start checkpoint. We're also feeding into the cyclin D. One way cyclin D effects the transition to the S phase is by inhibiting RB. But if we're inhibiting cyclin D, the retinoblastoma protein, you'll remember, is a protein which is binding the transcription factors that cause cell cycle progression. So retinoblastoma is active. You do not have cell cycle progression. And the logic here between activation and inhibition is encoded in this molecular wiring diagram, if you will. Now I'd like to highlight another type of pathway, so I'll type in KEGG, pathway lung cancer, and here we have a pathway that KEGG has annotated for what's going on, and here we see the EGFR receptor. This is a receptor tyrosine kinase, and also the protein ERB2, which is a related protein. You might also know this protein by the name HER2. It's one of the proteins that you target for breast cancers, but these will act as heterodimeric receptors, and these double lines are the membrane. Here we have the ligands which can bind to these receptors. And then when you look what's happening downstream, you can see a couple of proteins hitting RAS. This is the protein that's mutated in cancers. Frequently, RAS hitting the MAPK signaling pathway, and the MAPK signaling pathway will also interact on cyclin D1 and causing proliferation. Other downstream effects of this receptor is the inositol triphosphate signaling pathway, which again, this is a glycolipid with a sugar molecule on top of a lipid. It gets cleaved, the different molecules do different things, and it's going to impact the AKT-mTOR pathways, which is involved in a lot of cell growth activities. And we can get a more expanded view if we click on this link. Here you can see the principal role of AKT impacting a lot of proteins downstream involved with metabolism, cell cycle progression, cell survival. And what's perhaps a little less obvious is AKT's, unless you know what you're looking for, AKT's influence on the mTOR signaling pathway. And here we see the mTOR pathway is central towards protein translation and cell growth. So we've affected, the accumulation of mass, cellular division, and cell survival necessary for cancers to occur. So you can see that these wiring diagrams of the inside of cells can be pretty useful for understanding processes in a larger way. So KEGG is certainly one place to look at. The other source that perhaps is the sun is setting a little bit, but it was one of the main repositories for pathway information for a long time, was this BioCarta website. And if we type in P53, we can see its role in it has human pathways and mouse pathways. It might be slightly different. Let's check the human pathway. Again, here we have DNA damage affecting P53, P53 interacting through an intermediary to impact, well, a couple of different intermediaries, CDK2, CDC25, again, hitting cyclin D, the same thing that Keg was talking about, which is going to impact retinoblastoma and is going to influence the transition from G phase to S phase. They also have nice summaries of what's going on in the logic of the pathways underneath these. So these are two very valuable resources for looking at functions of genes in larger contexts of other proteins. Okay, thank you for listening.